Glucokinase versus hexokinase. Both catalyze the same reaction, conversion of glucose to glucose 6 phosphate, a kinase reaction, intermolecular shift of a functional group, phosphate in this case, from ATP to glucose to form glucose 6 phosphate. This is often referred to as first phase of glucose. After glucose enters the cell, the first phase is it is converted to glucose 6 phosphate and it commits the glucose to intracellular metabolism. Just a point why it is phosphorylated first of all. I have written here trap because glucose, the way it entered the cell, facilitated transfusion by carrier mediated. Glucose can as easily leave the cell also by the same transporter. But after it is phosphorylated, the phosphate group is too polar, is highly polar, negatively charged. So this does not cross the membrane. So it is trapped inside the cell. इंट्रासेलुलर मेटाबॉलिज्म का कमिटमेंट मैंने बात की थी इट कैन गो फॉर अ नंबर ऑफ pathways of course glycolysis in the fasting state etc into the gluconeogenesis again by a different enzyme glucose 6 phosphatase it can go for alternate pathways HMP shunt or the uronic acid pathway and in the fed state it can go for glycogen synthesis. What role these two enzymes, actually isoenzymes, glucokinase is type 4 isoenzyme of hexokinase. So before we come to different properties of this, which is aim of this lecture, let us consider one point. We humans are not continuous eaters. You take 2 or 3 major meals a day. So blood glucose rises at certain times of the day only. There is a rising tide of glucose. So it is required that this glucose, after conversion to glucose 6 phosphate, it should be converted to glycogen by the process of glycogenesis. In rest of the 17, 16, 18 hours of the day, glucose, even if it is the level is low, it should be converted to glucose 6 phosphate. And there an important statement when the blood glucose level is low, non-fasting, in the postprandial state, the glucokinase comes into action and channelizes glucose 6 phosphate to glycogenesis. Rest of the times it is hexokinase, it is operative. It can act on glucose even when the blood glucose concentration is low. Why? Because hexokinase has got high affinity for glucose. So it can phosphorylate glucose even when blood glucose level is low, even lower than 100. For example, in the fasting state, when it is falling down, even that time also, when it goes below 100 also, it is able to act. Whereas glucokinase, it has got low affinity. So this is reflected by high Km of glucokinase and low Km of hexokinase. So what I mean to say that glucokinase comes into action because of its low affinity or high Km. It comes into action only when blood glucose level rises, when there is rising tide of glucose as it happens after the meals. Vmax, glucokinase का Vmax is high, इसका low. Returning to that postprandial state when the blood glucose level is rising. So that time it is desirable that glucose rising tide, this whole glucose should be phosphorylated and should be channelized into the glycogen synthesis, glycogenesis. So its Vmax is high. Vmax का क्या मतलब है? Vmax means the maximum speed which can be attained. So it has got low affinity, so it comes into action quite late when the blood glucose rises high. But once it comes into action with its high Vmax value, it is able to phosphorylate whole of glucose into glucose 6 phosphate without any hindrance. What I mean to say, it is not subject to feedback inhibition by glucose 6 phosphate. Glucose 6 phosphate is inhibitor of hexokinase activity. But glucokinase is not subject to such feedback inhibition. So I summarize, rising tide of glucose, glucokinase comes into action, promptly converts it into glucose 6 phosphate. High Vmax helps that, and lack of feedback inhibition also helps that. One more point, what further helps is that glucokinase is induced by insulin, hexokinase is not. So in the postprandial state, all of you know, high blood glucose, rising blood glucose, it stimulates beta pancreatic cells. तो उससे क्या होगा? This is called glucose induced insulin response. So insulin level goes high and insulin induces synthesis of glucokinase. So that further helps. Glucokinase ka availability of molecules also rises. What about distribution? Glucokinase, it's wrongly written, sorry. Glucokinase is present in liver and pancreas beta cells. Hexokinase, it is present in all tissues of the body. That is understandable because glycolysis, HMP shunt, etc. we mentioned, they occur everywhere in the body. What about specificity? Glucokinase is specific for glucose. Hexokinase, it can act on fructose, converting it into fructose 6 phosphate. It can act on mannose, converting it into mannose 6 phosphate. So various features are varying. So we have tried to explain how these features are helping high affinity of hexokinase, low affinity, high Km of glucokinase means it comes late into action, high Vmax without any hindrance. तो जो phosphorylation शुरू हुआ लेट हुआ बट वो फिर होता ही चला गया. Induction by insulin further helps. One important point I would like to say here कि ग्लूकोज को ग्लूकोज 6 फॉस्फेट एंजाइम इंडक्शन ग्लूकोकाइनेज इंड्यूस हो गया बाय इंसुलिन एक्शन फास्ट. Now insulin is also stimulator of the key enzyme of glycogenesis, glycogen synthase. So this glucose 6 phosphate is effectively channelized into glycogen synthesis. So this is going beyond the topic today, but this is just to tell you it doesn't stop at first stage. ग्लूकोकाइनेज का इंडक्शन हो गया और ग्लाइकोजन के अंदर चैनलाइजेशन भी हो गया. तो ये हमने जैसे बताया था ग्लूकोकाइनेज नैरो स्पेसिफिसिटी एंड broad specificity of hexokinase. Why phosphate? Just some related questions which are asked in Viva, of course. Trap. What do we mean by trap? Glucose 6 phosphate cannot leave the cell. अगर ये ग्लूकोज में ही रह जाता, glucose could have left by the same transport protein. But glucose 6 phosphate is too polar, very polar, negatively charged. It cannot leave. Another point, there are no transporters for glucose 6-phosphate. They are isoenzymes of each other. Glucokinase is isoenzyme 4 of hexokinase. What are isoenzymes? Catalyze the same reaction but with different kinetic properties. So we have seen today how their kinetic properties differ and how that helps. Last, maturity onset diabetes of the young. Now, beta cells, pancreatic beta cells, they are glucose sensing cells. They sense the blood glucose level and accordingly they release insulin. That is called glucose induced insulin response. Glucokinase decreases threshold of the insulin release. Okay, glucokinase decreases threshold for the insulin release. तो अगर ग्लूकोकाइनेज में कोई म्यूटेशन हो गया तो ये थ्रेशोल्ड इंक्रीज हो जाएगा. देयर विल बी ब्लंटेड response. The so-called glucose-induced insulin response will be blunted. So learn more about MODY. Thank you so much.